Hello class. So today's topic is about microbial diversity, part 1. We're dealing with acellular and prokaryotic microbes. So let's start with the viruses. Okay, so viruses are extremely small and you need an instrument, a special instrument like electron microscope to observe the viruses. Also, uh, this viruses is under acellular. Okay, acellular. Another thing, viruses are not alive, and in order to replicate, viruses must invade live hosts. Uh, Viruses need live hosts in order to replicate, in order to survive. Okay. Also take note that viruses contains either DNA or RNA, usually, but in rare cases there is viruses contains both. But typically viruses contains only DNA, either DNA or RNA. Okay. And the simple structure of viruses. So it consists of protein coat, okay, protein coat or the capsid, no? And in the center or inside of the capsid or protein coat, you can this is the capsid, okay, and inside the capsid you can see the DNA or RNA, okay. And if we are referring to the capsid enclosed, DNA enclosed in capsid, so that is nucleocapsid rather, okay. So yeah, this is the basic structure of a virus. It's not complex compared to eukaryotic cells, no. So here's the origin of virus. So we have two main theories. The first one suggests that viruses existed before cells. So however, this view is considered less plausible as viruses require cells in order to reproduce. Again, since viruses need hosts to reproduce, so the idea or the theory of viruses existed before cells somehow it is neglected, or it's not generally accepted. Okay. Next, another theory about the origin of virus is that viruses originate from cells and represents degenerated or simplified cellular components. So from a cell, okay, virus originated, okay. No, there's cells that exist, and then from it, no, the virus were degenerated, okay, no. Determining whether viruses are alive is a complex question, no? Because we know that viruses are not alive, okay. It requires host to remain alive, no, to exist, okay. Again, no, since viruses are considered not living organisms, no, it is referred as acellular microbes or infectious particles. So we have this special type of viruses, okay. So we have these bacteriophages, okay. In this case, the virus we have a virus and we have a bacteria, okay. Let's set that as an example. So the virus infects the bacteria, and then it transfer some DNA from some material inside of that bacteria and then replicate, okay. So basically it happens here is they're just reproducing, okay. And that process is called lactic cycle, okay. And this one, lactic cycle. So here are the steps, no, that happened during elliptic cycle. So we have this, 5 steps. Okay. So the first one, we have attachment. Okay. In this stage, okay, the page attacks the protein, or let's say the bacteria. Okay. So it attaches. Next, penetration. So the pages, or the bacteria pages, injects its DNA to the inside of the bacteria cell. Okay. Next, biosynthesis. So the page genes are expressed. Okay. So resulting in the production of page pieces or parts. So it's time for them to cultivate. Okay, inside. It's cultivating inside or propagating. Okay. Assembly. Once the biosynthesis is done, okay, the page pieces or parts are assembled to create complete pages. Okay, they are now transforming into they are simply maturing inside, and once they are fully matured, proceed to the last step, which is release. So once they are already completed, the pages escape from the bacterial cells, okay, by lysis of the cell. So this one, just refer to this illustration, no? We have this attachment, and then it penetrates the DNA. Then inside, there's a biosynthesis that happened, and then part of biosynthesis is there's an assembly of those of those DNA, no? Those parts, and then once it's already matured, so it's time to release. So that's elliptic cycle. Okay. Next, so bacteria is a bacteria are single cell, rather, prokaryotic microorganisms that lack a nucleus and membrane bound organelles. Bacteria is also is considered as a cellular organism. Okay, it can live without hosts. They are found in almost all environment, like soil, water, air, and even within the human body. Some bacteria are harmless, some are beneficial, and some are pathogenic. Okay, some are like let's say an example of these are bacteria found in your gut. Okay, some are those good bacteria, some are bad. Okay. So characterizing this bacteria, okay, so we have many parameters. So those includes cell shape, okay, morphological arrangement, staining reaction, motility, colony morphology, atmospheric requirements, nutritional requirements, biochemical and metabolic activities. Okay, and then this pathogenicity, okay, and genetic composition. So let's start with the first characterization. So bacteria is characterized based on its cell morphology. Okay, when we say cell morphology, it refers to the shape, size, and the arrangement of bacterial cell. Okay, so common shapes are cocci, bacilli, and then spirilla. This one is spirilla. Okay. When we say cocci, it is a spear shaped bacteria. Bacilli, a rod shape, and spirilla, it's a spiral shape or vibrious. Okay, comma shape. Take note that bacteria is reproduced by binary fission. So from a parent, okay, it is just simply splitting apart. Okay, splitting. So the time it takes for one bacterial cell to split into two cells is referred to as the organism's generation time. So again, basically, if we're talking about binary fission in bacteria, so it is splitting of one cell. Okay, splitting of one cell or one bacterial cell. Okay, so as you can see here, so these are the following example of cell morphology of a bacteria, no? In this illustration. Okay, so we have diplococci, streptococci, and staphylococci. Okay, I'm sorry for sorry for pronunciation, but I'm trying my best. Okay, and we have tetrad, octad. So based on its appearance, okay, so that will be its corresponding name. Okay. Okay. Next parameters that to be considered when classifying or characterizing bacteria is the stain reaction. So in nature, okay, most bacteria are colorless, transparent, and difficult to see. Okay, so we have these staining methods used enable to see enable to see or examine the bacteria. So when we say stain reaction, it refers to the way a bacterium respond to a particular staining method. Okay, so okay, to make it simple, okay, you have, let's say you have a clothes, okay, and then you're eating pasta, okay, and then suddenly you spilled out the sauce, and then there's a stain in your cloth. Okay, so basically it is somehow similar to this staining method. Okay, we're adding color to the bacteria so that we can see it and we can examine the bacteria. Okay, so the most commonly used stain is called gram stain. Okay, so in this gram stain, okay, the bacteria is divided into two groups. So the first one, it is gram positive, where its color is ranging from blue to purple, and the next is we have gram negative. Its color is ranging from red to pink. Okay, so to give you some idea how this gram stain perform, so starting with a specimen. Okay, a specimen a specimen with bacteria, no? And then you will flood it with crystal violet solution. Okay, next after is you will rinse the slide, then flood it with iodine solution. Okay, and then as you can see, the bacteria will turn into blue to violet color. Okay, next you will rinse off the excess iodine and decolorize it with acetone. Okay, so some of the bacteria will become colorless, some have color. Okay, next is after decolorization, no? So you can see some of the bacteria will be no longer visible. Okay, then apply safranin counter stain. Okay, so in this stage or in this step, no, after applying that, you will see some of the colors will become red, or it turns red, or it turns red or pink. Then once done applying the counter stain, so you need to wash it, blot and dry it through air, and then all of the bacteria will have its corresponding color. Okay, so just focus in this part, okay? So a gram positive bacteria, okay, if you perform this gram stain, okay, a gram positive bacteria, its color is ranging from blue to purple, and then a gram negative bacteria, its color is ranging from pink to red. Just at least memorize or remember that part, okay? Next, motility. So motility, another characteristics that define a bacteria. So motility refers to a bacterium's ability to move, typically by employing or using the structure like flagella, cilia, or doing some gliding mechanism. Okay. So if a bacterium is able to swim, it is said to be motile. Bacteria unable to swim are said to be non-motile. So straightforward. Okay. So bacteria, it can move by using its flagella. Yeah, its tail, somehow its tail. Yeah, so-called tail. Cilia, it's tiny hair. Okay, cilia, or it's they can move using the gliding mechanism. Yeah, this one, they're gliding, they're sliding. As long as they are in the solid surface, they're just sliding. Okay, gliding. Next, colony morphology. So colony morphology pertains to the appearance of group bacterial cell growing together on a solid media plate. This includes their shape, color, texture, evaluation, and margin. A mound or pile of bacteria on a solid culture medium is known as bacterial colony. So this is an example. Okay, so it starts with a single bacterial cell. Excuse me. Excuse me. So it starts with single bacterial cell. Then after an hour, or let's say after 4 hours, okay, it multiplies. It grows exponentially. Okay, it grows exponentially. After 4 hours, from 1 it becomes 256. Then after 8 hours it becomes 65,000, and after 12 hours, okay, it has 17 million. Okay, 17 million cells. Okay, so again, if you find some mound or pile of bacteria, this is called bacterial colony. Okay. Next, atmospheric requirements. So as atmospheric requirements describe the type of air or oxygen a bacterium needs to grow. Some are aerobic. So when we say aerobic, it requires oxygen to survive. Some are anaerobic, so it can survive or grow without oxygen. Or some are facultative anaerobes. No, it can survive either oxygen exists or not. Okay. So let's focus here in the illustration. Okay. So obligate aerobes and micro aerophiles, they tend to survive when there's a oxygen present. So these obligate anaerobes and aerotolerant anaerobes, they tend to survive without oxygen. They don't need oxygen to survive. So excuse me. excuse me. So these facultative anaerobes, so it's either there's a oxygen present or not, they can still survive. Okay. Next, nutritional requirements. Okay, some bacteria requires nutritions, no, not just like us humans. Okay, we need nutritions. So some bacteria requires carbon, nitrogen, vitamins, and minerals in order to grow. Okay, so some they use nutrients to generally they use nutrients to make carbohydrates, to make amino acids, to make lipids, vitamins, growth factors, or to make RNA or DNA. Okay, next, biochemical and metabolic activities. Okay, so bacteria is categorized or characterized based on its biochemical and metabolic activities. Some bacteria, okay, produce chemical reactions, okay, some bacteria produce chemical reactions in order to grow, reproduce, and produce energy. Example of this is fermentation, respiration, and the ability to break down complex compound. So, in short, bacteria, okay, bacteria is used, okay, it is used in fermenting food. No? Somehow, okay, bacteria are good. Let's say there's a time that when we hear bacteria, it's always bad. It causes disease. But in some cases, bacteria is helpful in our daily life. Like for example, fermenting food, in a let's say in cheese manufacturing, blue cheese, milk processing, milk. When milk turns into sour, so it tends so basically you have yogurt, okay, because there's a bacteria inside of it. And by the way, okay, so some bacteria excreted toxics, okay, and this refers to the pathogenicity of the bacteria, another characteristic of bacteria. So when you say pathogenicity, it refers to the bacterium's ability to cause disease in the host. Okay. Okay. So in some cases, we have bacteria inside our body, but still we can't get ill because we have strong immune system. Okay, with the process of phagocytosis, our white blood cells, the macrophages, the neutrophils, they engulf and destroy the bacteria. Okay, so basically some of our bacteria are dormant, or they're not spreading too much because of having an active immune system. Okay, so in short, okay, bacteria can cause disease. Okay, it depends on its classification. Okay, but some are good, some are beneficial. Okay, so the way they release toxic is part of their metabolism. Okay, or biochemical activity. So somehow this biochemical metabolic activities is somehow connected to this pathogenicity. Okay, next, genetic composition. So bacteria, okay, when we say genetic composition, we're dealing with the DNA of a bacterium. Okay. Typically, this DNA is a circular chromosomes along with plasmid. So based on its DNA genetic composition, bacteria is classified. Okay, some DNA causes illness, some are not. Okay. So we're done with the bacteria. So let's proceed to archaea. Okay, Archaea, Archaea is a prokaryotic organisms in the domain Archaea were first discovered in 1977. Originally, it is called Archaebacteria, but as time passes, okay, as we have further development, understanding about biology, so this Archaea is different from a true bacteria, okay, due to its unique genetic and biochemical traits. So the word Archaea means ancient, and so basically, some scientists tell us that it appeared very early in the Earth's history. Okay, but some scientists, but still, this statement are debatable. Okay, whether the bacteria or the Archaea came first. So another thing, Archaea are extremists or extremophiles, meaning they can live in a harsh environments like high temperature, high salinity, or low water conditions, as well as oxygen level, no? Having low oxygen level. Okay, so that's all. Okay, so that's our part 1 in microbial diversity. So next discussion is about eukaryotic and microbes. So thank you for listening.